Hi guys, welcome to our embryology series. Today we're going to talk about the development of the eye. Right, so let's get into it. Right here you see this is your embryo around at the end of fourth week of development. You can see two things that are very important as far as sensory organ development is concerned. Right here is your optic vesicle and right here is your aortic vesicle. Right. So your optic vesicle is going to be involved in the development of the eye, while your aortic vesicle is going to be involved in the development of the ear. Let's take a look. Now, your optic vesicle, it actually developed from the forebrain, right? Your diaphragm, right? Which is your forebrain. So I want you to take a look. Let us take a look at this. So right here, this is the wall of your forebrain. Around day 22, you're going to form what called an optic groove. These optic groove, they are bilateral. This one and this one. This optic groove will continue to invaginate, right? Um, and continue to invaginate and grow and forms this what you call an optic vesicle. Right. So this optic vesicle is actually an extension of the forebrain. Remember, the forebrain comes from the ectoderm that has undergone neurulation, forming a neural plate, and that neural plate will form a neural tube after folding, and then from a folding. At around day 28, you're going to have the closure of your cranial neural pole, and then from the close of the cranial neuropole, you're actually going to have this invagination, which is called the aortic vesicle, right here, the aortic vesicle. So the aortic vesicle is going to grow towards the surface ectoderm, right? And then it's going to induce the surface ectoderm to proliferate, right? And form a layer that is called the lens placode. The lens placode. So the lens placode, it is actually induced by the optic vesicle, which is coming from the forebrain. Right. So take note of this. The optic grooves, they are outpocketings of the forebrain around day 22. Day 22. Right. So right here, because of the induction of the optic vesicle on the surface ectoderm, we are going to have our lens placode. This lens placode will invaginate, right, we inside your optic vesicle, so that it will form, it will transform an optic vesicle to an optic cup. Let's take a look at another diagram. Right. So this is an optic cup, right? So an optic cup is going to be suspended to the forebrain by an optic stalk. So you have an optic stalk and then you have an optic cup, right? This was once called an optic vesicle. But because of the invagination of the surface ectoderm, which you call the lens placode, you're going to form a groove within this optic cup. Right. So take note of this. The optic cup is going to be C-shaped in cross section. C-shaped in cross section. It's going to have two layers, an inner layer and an outer layer. And between the inner layer and the outer layer, you're going to have what called an interretinal space, an interretinal space right here that's going to initially separate the inner layer from the outer layer. But take note of this. This optic cup is actually going to is actually going to it is not going to grow inferiorly or proliferate inferiorly. Therefore, there's going to be an opening in its inferior aspect, which is called a choroid fissure. A choroid fissure. So this is your choroid fissure, this is your choroid fissure, and this is your choroid fissure right here. So this is a longitudinal section, and you can actually see that inferiorly it is open. Why is it open? Right. So there's actually going to be an artery that is called a high a hyaloid artery. This hyaloid artery will actually be involved in supplying blood to your developing lens, your developing lens. So you can actually see this is your hyaloid artery, right? Embryologically, this hyaloid artery will extend right towards the lens vesicle, but postnatally you are going to have a part of the hyaloid artery which is within this stalk, which you call the optic stalk, and then a part is going to extend to the lens vesicle. So therefore it's going to be divided into two. You're going to have a proximal part and a distal part. And NB, the distal part of the hyaloid artery is going to turn, is going to obliterate and form the famous hyaloid canal. So you can actually see this in the cross section or in the structure of an adult eye. But the proximal one, which is very important as far as the retina blood supply is concerned, you're going to have what's called the central artery of the retina. That's the proximal portion of the hyaloid artery. Very important. All right. So we say that you're going to have your forebrain. You're going to have your forebrain forming out pocketings of your called your optic growths around day 22. These they will grow and fuse with the surface ectoderm, induce the surface ectoderm to proliferate to enlarge, and then forms what you call a lens placode. This lens placode will invaginate into your optic vesicle, forming an optic cup. But this optic cup is going to be deficient inferiorly, forming what you call a choroid fissure for the passage of the hyaloid artery. Very important. Right. Right. So take note of this. That the choroid fissure is going to close around week 7. So week 7, right, you're going to have the inferior portion of your choroid fissure actually closing. You understand? So this is going to close around week 7. Very important. So you're going to be left with a secular, a secular space. Because if you close this, you're going to have a secular space. That secular space is going to be your future pupil. Future pupil. Very important, right? Good. Right. So let's take a look. What happens to the lens placode after the optic vesicle is fused with the surface ectoderm? Let's take a look. Right, so this is your forebrain, and this is the outpocketings that we are talking about. These are your optic vesicle. This optic vesicle, they will contact with the surface ectoderm and induce the formation of the lens placode. Right. This lens placode, they're going to invaginate inside. Let's take a look at another diagram that actually reveals the invagination of the lens placode. So this lens placode will continue to invaginate and invaginate up until it loses its attachment from the surface ectoderm. That actually happens during week 5. So during week 5, that's where you have the lens placode losing its attachment from the surface ectoderm. Let's take a look at the diagram. Right, so this is actually showing the growth of the lens placode. So this is the lens placode. It is vaginating, forming a lens pit, right? And then the pit will lose its attachment from the surface ectoderm and forms your lens vesicle. And take a look that the lens vesicle is going to be surrounded by the optic cup, by the optic cup. So this lens vesicle is the one that's going to be responsible for forming your lenses, your lens, right? Right, so it's going to form your lens, right? And also it's going to form the corneal epithelium that will be surrounding the superficial surface of the cornea. That is your stratified non-keratinized squamous epithelia that surrounds the outer surface of your cornea. It actually comes from the lens placode. You understand? Good. Right, so let's take a look at the development of the lens itself. The lens itself coming from the lens placode. So the lens is going to be surrounded by these cells that I have come from the surface ectoderm. These are actually your cuboid, your cubic, they're just cuboidal cells, right? So this is going to be your posterior wall of the lens, and this is going to be your anterior wall of the lens. And take note, the cells within the posterior wall, they are actually going to elongate anteriorly up until they will fuse. So these ones, they'll be growing and growing and growing up until they fuse with the anterior wall. Right. So take note of this. The posterior cells, they will reach their anterior wall around 7th week. The 7th week, around 7th week, these cells, so they'll move from being cuboidal and then they become columnar as they reach the surface ectoderm. So as they reach the anterior wall of the lens, like this, right? And then these are the cells that will later form your lens fibers. Remember, your lens is made by lens fibers. These are not actually fibers, but they are actually cells which are called lens fibers that you have crystalline, that is actually transparent, responsible for refraction of your what? Of your light. Is it? Very important. Right, so let's take a look at the development of the retina. So the development of the retina is actually complicated, but I'm going to summarize so that you may understand. It's very simple. So remember, we said we have an optic vesicle that is going to evaginate from the forebrain, goes towards your surface ectoderm, that is going to form a cup due to the invagination of the lens placode that will later become your lens vesicle. Importantly, this optic cup is going to be made by two layers of cells. You're going to have what's called an inner cell layer, this one, right? And then you're going to have an outer cell layer, this one. And between the outer and the inner cell layer, you're going to have your intraretinal space. This interretinal space is very important because they are actually, you're going to find cells within this interretinal space, and these cells, they are actually going to be responsible for the formation of all those layers within your retina. And you remember from histology, we understand that the retina is made by 10 layers, and the most outer layer, very important, is going to be the pigmented layer of the retina. So the pigmented layer of the retina is going to be formed by the outer layer of the optic cup. So this layer is going to form the pigmented layer of the retina, right? And then right in your intraretinal space, there are cells that are called your that are found in the posterior 4/5, right? This area in blue, so this area in yellow, right? So the posterior 4/5 cells, these are called your pars optica retinae. So your pars optica retinae, these cells, they are going to be responsible for the formation of your rod and cones. Your pars optica retinae are going to be responsible for forming your rods and cones. They make up the posterior 4/5 of the inner layer of your optic cup. Just interestingly, you have to understand that the rods and cones, they are called the photo sensitive receptors of the eye. And importantly, the rods, you have about 120 million rods and 6 to 7 million of cones. So therefore, the rods are more numerous as compared to cones. The rods are more numerous and they are even more sensitive compared to cones, but they do not detect colored light. They are for black and white. Black and white. The cones are the ones that are responsible for differentiation of colors. So these, they are coming from your pars optica retina, which is your 4/5 of the inner layer of your optic cup. Right. Let's take a look at the anterior 1/5 of the inner layer of your optic cup. What does it forms? Right. So this anterior 1/5 is going to be called your mental layer. So the mental layer is actually going to is the same mental layer that is found in the development of the brain, right, which will develop your neuroglial cells and involved in formation of some supporting tissue. So the anterior one-fifth of the inner layer, which is called the mental layer, is actually called the pars ceca retinae. So that pars ceca retinae, the pars ceca retinae, right, is actually going to divide into two and form two layers of cells, right? So it's going to be responsible for the formation of the following. Take note: the anterior one-fifth will form your iris, mostly your inner layer of the iris, right? And it's going to form your ciliary body. Remember your ciliary body with your ciliary muscles, with your ciliary zonules, and so forth. You understand? So you're going to have your 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 anterior one-fifth, which is your pars ceca retinae, giving rise to your pars iridica retinae. Your pars iridica retinae is the one that will form the inner layer of the iris. So you're going to have your iris, so the inner layer of the iris, and then you're going to have your pars ciliaris retinae that will form your ciliary body. Let's take a look at another diagram that is more visible. Right, so this right here is your ciliary body, which will form your ciliary muscles that will be responsible for the accommodation of the lens and so forth, right? So this is coming from the anterior one-fifth layer, which is called your pars, right? Ciliaris retinae, is it? And then this layer right here is going to form your inner, this layer in blue, in yellow, right? It's going to form your inner layer of the iris. That's your pars iridica retinae will form the inner layer of the iris. So let's take a look at a revision of the development of the retina. Right. So the retina, we say it comes from the inner layer of the optic cup, right? Divided into two. You have your posterior four-fifth, that's your pars optica retinae, that will form your rods and cones, this whole layer, right? Rods and cones, right? And then you're going to have a short layer anteriorly, which we call the pars ceca retinae, which will be divided into two. That's your pars ciliaris retinae and your pars iridica retinae. Pars ciliaris retinae will form your ciliary body. Your pars iridica retinae will form your inner layer of the iris. Good. And this is your lens. You can see the posterior cells of the lens. They will tend from being cuboidal to be longitudinal or to be columnar, reaching the anterior layer of your lens. These will later become your lens fibers. And we say they will reach the anterior portion by week 7. By week 7. Right. So let's continue. Right. So anterior to your lens, right, you're going to have um, two compartments, very important. You're going to have an anterior chamber, and then you're going to have a posterior chamber. So you're actually going to have loose mesenchyme that will vaginate, separating your lens, right, to the surface ectoderm. Right, remember the lens became the lens placode vaginated and become the lens vesicle, and that the space between the surface ectoderm, right, and the lens, right, you're going to have your anterior chamber and your posterior chamber, right. So your anterior chamber, which you will be filled with loose mesenchyme, right, will actually form two epithelia, right? It's going to form two epithelia, right? The anterior epithelia of this loose mesenchyme will form the endothelium of the cornea, the endothelium of the cornea, right? And then the posterior layer of the mesenchyme will form what's called the iridocorneal membrane, which is also called the papillary membrane, right here, right? So this papillary membrane, it will later obliterate. Take note of this. The papillary membrane or the iridocorneal membrane will later obliterate. That is the posterior a membrane formed by the loose mesenchyme between the surface ectoderm and your lens, right? Take note of this. The one that is anterior, so the mesenchyme in this region is going to form the endothelium of your cornea, the endothelium of your cornea. And then the space that is between uh, um, within, okay, best that we are talking intra-embryonic life, in intra-embryonic life before the obliteration of the iridocapillary membrane. The cells that are within here, this space, they are going to be filled with fluid. That is what you call your vitreous fluid. So this vitreous fluid will form the anterior chamber. Will form the anterior chamber. You understand? Good. Then between the iris and the lens, that's where you're going to have your posterior chamber, right here. Right. So initially you're going to have your papillary membrane separating your anterior chamber from your posterior chamber. When it obliterates, you're going to have open communication between your anterior chamber and your posterior chamber. This communication is via the pupil, and it's very important as far as a nutrient supply to the lens, to the avascular lens and avascular cornea is concerned. Right? So fluid of the posterior compartment will be moving to the anterior compartment via the pupil, right? And then from the anterior compartment, this fluid is going to be drained, right, by a canal called the canal of Schell, or the sclera of venous sinus. Um, that will later be connected to your veins, right? So you're going to have the fluid in the anterior chamber being drained and so forth. Right. So let's take a look at the development of the cornea. We already spoke. The cornea, remember, is made up of 5 layers histologically, but embryologically, we speak of just basically 3 layers. Right. So the surface ectoderm, the surface epithelium of the cornea comes from the ectoderm. Is the ectoderm surface that is going to be stratified squamous, stratified squamous non-keratinized epithelium right here. Good. Coming from the surface ectoderm. The stroma of the cornea, which is also called the substantia cornea, right? Sorry, which is called the substantia propria, right? Is the one that's going to form your stroma. And remember from histology, the stroma forms 90% of your cornea. And the stroma is actually the extension of your sclera, as we're going to talk about very soon about the sclera and the choroid. So the extension of the sclera anteriorly will form the substantia propria, right? Substantia propria, which will become the stroma of the cornea. And then the last layer of the cornea, that's your inner layer, which is your endothelial layer, which is formed from the loose mesenchymal layer that was between the lens and the surface ectoderm. Right. NB, very important. This one you have to take note of this. The loose mesenchyme between the iris and the cornea, right, coming from the ectoderm, will form the muscles of the iris. That's your dilator papillae and your sphincter papillae. So an MCQ question can come and says, your sphincter papillary muscle, you have endodermal origin. That is false. They are ectodermal origin because they are formed by the mesenchyme coming from the ectoderm. So your sphincter papillary muscle together with your dilator papillary muscle, they are coming from the ectoderm. Very important. Right, so let's take a look at the development of your fibrous layer, your vascular layer. We spoke of the neural layer, right? So remember, we said that the optic cap is coming as an outpocketing of your forebrain. The forebrain around the neural tube, it's going to be surrounded by pia mater, and then from pia mater, it's going to be surrounded by, what you call So pia mater is going to be surrounded by pia mater and it's going to be surrounded by dura mater. So the extension of the pia mater coming from the development of the brain, that extension of the pia mater is going to extend as the choroid. So the choroid is the extension of the pia mater. You understand? That's going to be your vascular layer. As for the sclera, the dura mater, right? Remember the leptomeninges that are formed, that is the layer around your brain, right? Which is involved with the pia mater and the dura mater, right? So the dura mater of the brain will form the sclera, which is your fibrous layer of your what? Of your eye. And then we said the sclera anteriorly, it will become your substantia propria, which will become the stroma of the cornea. Very important. All right. So this is what is important that you have to take note as far as the eye development is concerned. Right here, this is your hyaloid artery, which is said distally is going to obliterate, forming the hyaloid canal. But proximally is going to continue as the central artery to the retina, which is going to supply most of your blood supply to the retina. You understand? Good. Let's take a look at some clinical correlates. Right. So here we have two things that we have to understand. Right here, your option A, that's your coloboma. That's coloboma iris, right? So what is coloboma? Remember, we said around seventh week you have the closure of the choroid fissure, right? That was the passage of the hyaloid artery. So if the choroid fissure fails to close, you're going to have an opening which is called which will later become as coloboma, right? So this is going to be the opening of your iris. It's going to extend your sclera, your choroid, all right, and actually um, uh, be involved with a lot of um, eye 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 abnormalities. You understand? So this coloboma, and then to our right here we have a persistent irido papillary membrane, which we said is going to obliterate, right? So if it just fails to obliterate, you actually have a layer of membrane that is just anterior to your pupil, all right? Good. Let's take a look at another clinical call. Right, so right here it's another abnormality which is called synophthalmia. So synophthalmia is just due to your eyes, they are going to be fused, right? So remember, we actually have some midline structures that you have to develop your nose from those frontonasal sinus, frontonasal prominences, all those things. So if they fail to develop, you're actually going to have your right and your left eye being fused together. That's your synophthalmia. And then lastly, let's talk about aniridia. So aniridia is just the absence of your iris. So you actually have no color, those beautiful color, blue eyes, brown eyes. You don't have those when you have aniridia. So that's absence of your iris. And then most importantly, another one is eye close, is what we call glaucoma. So if you remember very well, I said that the fluid which is found in your anterior chamber is going to be drained in the canal of Schell. So if there is a failure of that drainage of that fluid, it's going to accumulate. As it accumulates, it can actually compress the retina, compress the optic cord or optic nerve per se, and that can lead to blindness. That is what you call um, glaucoma. So thank you so much, guys, for attending this lecture. See you next time. Take care.